Hello friends, welcome to today's session regarding cell cycle and cell division. Myself Jalaja Sajeev, PGT Bio from KV Keshapuram. In the last session, I have given you some of the questions to think and respond. Now, you can see and verify your answers. What is the average span of cell cycle in mammalian cell? So, here you can check your answer, about 24 hours. Now, see the second question. Distinguish cytokinesis from karyokinesis. Yes, you know the answer. Cytokinesis, division of cytoplasm. Karyokinesis, division of nucleus. Karyon means nucleus and kinesis means division. So, karyokinesis is division of nucleus. Now, see your third question. Describe the events takes place during interphase. You know, interphase is the first stage of cell cycle and it consists of different phases. Yes, first one G1, then second is S phase and third is G2, then the fourth one, that is M phase. So, it consists of G1, S, G2 and G1, S, G2 we will collectively called as the interphase. Now, come to the next question. What is G0 of cell cycle? G for gap and 0 means here it is a quiescent stage, inactive stage. When cell becomes inactive, it will stop the cell division and it will enter into the stage called as G0 stage or it is known as quiescent stage. These cells, the cells that have already entered in this G0 phase, they can enter into G1 stage again and they can start the normal cell division if there is any requirement. So, as per the requirement, the cells that are entering into the G0 phase or even the quiescent stage, they can continue further cell division. Now, why is mitosis called an equational division? Equational, equal, equal in all respect, equal in quality and equal in quantity. So, when after this particular mitosis, the two daughter cells formed and these two daughter cells, they are qualitatively and quantitatively remains the same. Hence, it is known as equational division. So, they will be in all aspects similar to the parental cell. Name the stage of a cell cycle in which the following events takes place. The first one, chromosomes are moved to spindle equator. In mitosis, in which stage? The chromosomes, they are moving to the equator. Equator means center of the cell, middle portion of the cell. So, in which phase this movement of chromosomes takes place? Your answer is correct. Yes, it is the metaphase stage. Now, the second question here is centromere of a particular chromosome that will splits and the chromatids, the sister chromatids, they will separate. So, in which of this stage this event takes place? Yes, of course, your answer is again correct. It is the anaphase stage. So, during the anaphase stage, the centromere of each chromosome will divide and the sister chromatids of each chromosome that will move to, they will start moving to opposite poles by the contraction of spindle fibers. Can there be mitosis without a DNA replication? If there is no DNA doubling, then is it possible? In order to produce two equal cell, qualitatively and quantitatively similar daughter cells without any DNA replication, is it possible to occur? Yes, your answer is no, not at all. It is not possible. So, there won't be any mitosis without any DNA replication, because DNA replication is highly essential. Without DNA replication, we can't produce two daughter cells equally. So, for the production of two equal daughter cells, qualitatively and quantitatively similar daughter cells, before this division there needs DNA replication. Now, your question is, how does cytokinesis of a plant cell differ from that of an animal cell? Yes, you can verify your answer. Cytokinesis in the case of animal cell. So, before that, what is cytokinesis? Kinesis again means division, division of cytoplasm, that is known as cytokinesis. And in animal cell, you know, it is by which phenomenon? Yes, it is by cell furrowing. The plasma membrane, the outermost boundary in the case of an animal cell, it will produces invaginations, infoldings on either side of the cell in the middle portion, and later these invaginations they will deepens, this furrow that deepens and it will meet together and divide the cytoplasm equally into two. So, by this method the cell division and cytokinesis that is completed in the case of an animal cell. So, it is by cell furrowing. Now, in the case of a plant cells, how this phenomenon takes place? So, it is by cell plate formation. Now, how the cell plate is formed? What are the components involved in it? Fragments of endoplasmic reticulum, then vesicles of the Golgi bodies, and even the fragments that are formed from spindle fibers. All these they will arrange together at the center of the cell or even the equatorial plate of the cell, the middle portion of the cell. And these things, all these things are collectively called as phragmoplast. Later, all these phragmoplast, they will unite together and they will form the middle lamellae. So, it will fuse together and it will unite with on either side of the cell and it will form a layer at the center. So, it is known as middle lamellae. And on either side of this middle lamellae, then the cellulose materials will be deposited and it will form the cell wall. So, by this method the cytoplasm of the cell, plant cell, that will divide equally into two. So, it is by cell plate formation. Now, coming to the meiosis remaining aspect. we have already covered the first aspects regarding the meiosis. What is meiosis? What is its significance? Where it happens and why it happens. So, what is meiosis? Get a quick feedback. Meiosis means reduction division. Why it is called as a reduction division? Because the chromosome number is, yes, reduced it to half, and it takes place in a diploid cell. It takes place only in a diploid cell in order to reduce the chromosome number equally half in all the resulting cells. And in meiosis, it takes place in which cell? In which cell it takes place? It takes place in the reproductive cells or in the germinal cells. And by meiosis, we will get 4 daughter cells, because the nucleus will divide twice. There are 2 nuclear division, meiosis I and meiosis II. So, we will get 4 daughter nuclei, and each daughter nuclei are haploid with only one set of chromosome. And why it happens? It happens to bring the chromosome number to half, reduce it to half. It helps in sexual reproduction, and it results in the formation of gametes. So, for this purpose there needs meiosis, and it takes place in the germinal cell. So, as I have already told you, there are 2 divisions, meiosis I and meiosis II. And here each of these stages, again same, prophase I, metaphase I, anaphase I, telophase I. Then after that there will happen the cytokinesis. It is not necessary that immediately after anaphase and immediately after telophase I there needs cytokinesis. It may or may not happen. Then after that there will happen the meiosis II with all the substages. So, first meiosis I, that is the first division, meiosis, the nucleus will divide first. So, that division we will call it as meiosis I, first nuclear division, and it is having different substages. What are the different substages? Prophase I, metaphase I, anaphase I and telophase I. Now, coming to the first phase, that is the prophase I, and this prophase I, you see it is a longest stage in the meiosis I, and it has 5 different substages. The first substage, that is leptotene. Leptotene means thin thread stage. I will show you the diagram here. You can see the cell with these centrioles, with astral rays, and here the chromosomes you can see in the form of a thread like structures only, and this particular stage we will call it as thin thread stage. Chromosomes are seen in the form of ultra thin threads. We can't count the number of chromosomes, they are not distinct, they are not clear. So, this particular stage, the initial stage in prophase I, that is called as the leptotene stage. So, leptotene, teen means thread and lepto means thin. So, thin thread stage, leptotene is known as thin thread stage. Size of this nucleus will increase and formation of aster starts. Nucleus size is increasing because synthetic activities are going on, and aster formation will start from the centriole. coiling and condensation of chromatin reticulum will takes place. Again the chromosomes here we can see it as a network like structure, thin threads. So, these thin threads they will undergo coiling and condensation. Each chromosome has 2 sister chromatids, but that is not clear, but they will appear as single thread like structure, even though it is in doubled condition, but it appears as a single thread like structure. So, this particular stage, thin thread stage, that we will call it as leptotin, and it is the first stage of prophase I. Now, here you can see the second stage, zygotin. Zygotin means pairing of homologous chromosomes takes place. You see this diagram, you can see it clearly here. When you are observing here, you can see nucleolus is present, and these are the homologous chromosomes. This red and green chromosome that I have shown here, it is the homologous chromosome. So, here we can see 2 pairs of homologous chromosomes. Again you see the chromatids here, they are appearing as threads only, because this process of coiling and condensation that will that is undergoing, and it is seen only as a thread like structure. And here you can see the centrioles, doubled centriole, duplicated centriole with the aster formation here. So, this particular stage where you can see these homologous chromosomes are always coming together and they are seen as pairs, and this particular stage, that is the pairing of homologous chromosomes, that is known as zygotin stage. And this particular pairing of homologous chromosomes during the zygotin stage, that is called as synapsis. So, this stage zygotin is known as synaptic stage. The homologous chromosomes, they undergo pairing, and this pairing process we will call it as synapsis. Here in this diagram also you can see these 2 chromosomes, the one blue and the other one pink or even magenta color, this one, these 2 they are the homologous pairs of chromosomes, identical chromosomes, homologous. They are morphologically similar in its centromere position, length of the arms and all, it is similar. So, such identical chromosomes, they are known as homologous chromosomes, and these homologous chromosomes they pair during this particular stage, and that pairing of homologous chromosome, that is known as synapsis. Now, after synapsis, when you are observing, you can see that they are a group of 2, and this group of 2 chromosome we will call it as bivalent. Valency shows number and bi means 2. They are 2 chromosomes, but they will seen as a single pair. So, we will call it as bivalent. Now, coming to this one, this in a bivalent, you can see one chromosome is coming from the male gamete, that is known as paternal chromosome, and one from the female gamete contributed by the female parent. So, that is known as maternal chromosome. So, one is maternal and other is paternal. Paternal refers to father, contributed by the father, and the chromosomes contributed by the mother, maternal chromosomes. So, there are a bivalent has a maternal chromosome and a paternal chromosome. Now, these chromosomes again undergo coiling and condensation. This is a continuous process. It will take place till metaphase I. And after this pairing, that means after synapsis, you can see a complex structure, and this complex structure which is known as synaptonemal complex. And in this synaptonemal complex, you can observe one maternal and one paternal chromosome. Now coming to Packettine stage. Packettine stage is known as crossing over stage. See the diagram here. Again you can see clearly the changes taking place. Nucleolus is intact, it is present clear, and it is, you can see it clearly. These are the centrioles, duplicated centrioles, they start moving apart. There is the formation of asters, astral rays, and here the bivalent. This is a bivalent, a group of two chromosomes, homologous chromosomes. Now see what happens here is these chromosomes of this bivalent, and they exchange the segments of their chromatids. See these two chromatids, here you can see two red chromatid and two green chromatids. So the two chromatids of a single chromosome, that is known as sister chromatid. The two chromatids of two different chromosomes, that is known as non-sister chromatids. Now here in this particular diagram you can see these non-sister chromatids are exchanged. They are exchanging their segments, and this particular phenomenon, that is known as crossing over. So this crossing over takes place during Packettine stage. The point of crossing over, that is known as chiasma. So it is known as crossing over stage. Which stage is known as crossing over stage? Packettine stage. Paired homologous chromosomes, they undergo crossing over. Paired homologous chromosome means bivalent. Bivalent, they show crossing over. Further coiling and condensation takes place. So chromosomes becomes thick and rod shaped structures. Then here already I have told you each chromosome has two sister chromatids. The two chromatids of a chromosome, that is known as sister chromatids. And they are joined at the center. So we can call them as dyad. Each bivalent has two dyad. Di means two. A chromosome has two chromatid, so we will call them as dyad. This is yet another chromosome with a dyad, the two chromatids. So this is another dyad, this is yet another dyad. So a bivalent has two dyads, so we will call it as a tetrad for stranded stage simply. Now, what is the definition of a crossing over? See this diagram here clearly. It is exchanging the segments of non-sister chromatids. So, this phenomenon, that is known as crossing over. That means exchange of segments of non-sister chromatids of a homologous chromosome. It is known as crossing over, and the point of a crossing over is known as chiasma. Now, number of a chiasma indicates number of crossing over. Now, what is the need for this? It helps in the exchange of genes between the chromosomes. It helps in genetic recombination. It helps in variation of characters. It plays an important role in evolution. It helps in the exchange of genes So, it will definitely result in variation of characters which contributes to evolution. Now, coming to the next stage, that is known as diplotin stage. Diplotin is known as terminalization stage. Here you can see the diagram. It is known as terminalization stage. Terminal means moving towards the end, moving towards the tip portions. So, here this chiasma, that is moving, and it will reach even up to the end of the chromosome, up to the terminal portion. So, this even though it is moving to the terminal portion, these paired chromosomes, they will remain as groups. So, moving a part of chiasma towards the terminal, that is terminalization. So, it is diplotin is known as terminalization stage. Remaining characters, this centriole, duplicated centriole, they started moving and they are almost reaching the opposite poles. Astral rays formation is there and nucleolus is also present, but starts disappearing, disintegrating. Chiasma move towards the end of the chromosome, to the terminals. Asters move towards the opposite poles of the cell. Nucleolus starts to disappear, starts to disintegrate. Nuclear membrane will also starts to disappear, not totally disappeared, disappearance is starting. Now, the next stage, that is known as the diakinesis stage. Here you can see the diakinesis stage. It is known as repulsion stage. Here totally the chiasma is moved apart. You can't see any chiasma here in this pair. Even after that also the bivalents, they will remain together. So, this particular phenomenon we will call it as diakinesis or repulsion stage. Here bivalents will appear as ring like structure. There, even after repulsion they will remain as a single group. So, it appears as a ring like structure due to repulsion between the homologous chromosomes. Nuclear membrane disappears completely. Nucleolus also disappears completely at this stage, and spindle fiber formation is almost going to complete. So, metaphase 1. Prophase 1 is over with 5 different substages. You know what are the different substages? Leptotin, zygotin, pacutin, diplotin and diakinesis. Now, coming to the next stage, that is metaphase 1. Here you can see these bivalents, that means a group of 2 chromosomes, they are seen arranged at the center of the cell. See the diagram. Bivalents, maternal and paternal chromosomes, they are seen arranged at the center. This is one pair of chromosome, one pair of homologous chromosome, and this is the second pair of homologous chromosome. And they are seen arranged exactly at the equatorial plate of the cell. So, this particular stage, this is known as metaphase 1. So, bivalents are arranged at the equatorial plate of this cell in parallel groups. They are arranged parallelly. They are seen as parallel groups only. Centromere of the homologous chromosomes are pointing towards the poles. See the diagram clearly. You can see that they are pointing towards the poles and arms towards the center. Arms are lying at the center position and poles are pointing towards the centromere is pointing towards the poles. So, this particular arrangement that we can see here in metaphase I of meiosis. Chromosomal fibers, chromosomal fiber, the spindle fiber that attaches with the chromosome, that is known as chromosomal fiber. The chromosomal fibers attaches with the centromere of the chromosome. You see, here you can see the centromere and the chromosomal fibers, these are the spindle fibers, they are attaching with the centromere of the chromosome. Now, next is anaphase I. Spindle fiber will contract. After that contraction of a spindle fiber, you can see here in this diagram, just go through the diagram. Here you can see the separation of a bivalence. A bivalent, it is a group of two chromosome, homologous chromosome, and they are separating. The members of a bivalence are separating, not the chromatids. This is a very important point to be cleared here. So, the bivalents are separating and they are moving to opposite poles. So, in each pole you can see the member of one member of each of the bivalent. So, this happens by the contraction of spindle fiber. The chromosomal spindle fiber, it will contract and there happens the separation of bivalence and separated bivalence. What term we will say? Univalent. When they are seen together, we will call it as bivalent. But when they are separating, then we can say them as univalence. So, separated bivalence, that means the univalence, single chromosome that will move it to opposite poles. So, chromosome number reduced it to half in each pole. In each pole you can see the reduction of chromosome from 4, now it goes to 2 only. So, chromosome number is reduced. The actual chromosome number reduction that takes place in anaphase I. Centromere of univalence point towards the poles. You can see here the centromere of each of these chromosome, each of these univalent, that is pointing towards the poles, and arms are pointing towards the center. Now, coming to telophase. Here you can see it is just opposite to that of the prophase I. All the changes that takes place in the prophase I, that is just the opposite here. See, chromosomes, that means hereafter we will say it as univalence, undergo uncoiling, not coiling, uncoiling and decondensation to form chromatin reticulum. See this particular diagram here, each of this univalent undergoes uncoiling and after that decondensation, so that they forms the chromatin reticulum. Now, in this stage again this nucleolus reappears, and here there is only the centriole that will disappear. So, coming to the different points, chromosomes or univalents undergo uncoiling and decondensation to form the chromatin reticulum. Spindle fibers and astral rays, no need, they will disappear. And centrioles surrounded by a clear area, they forms the centrosomes. Centrosomes are formed and we can see one haploid nucleus in each of the pole. So, this particular stage that we will call it as telophase. Now, coming to cytokinesis 1. It is the division of a cytoplasm into two, equally into two, in two daughter cells. And in animal cell, it takes place by self-harrowing. Again the same method, no difference, just like that of the cytokinesis in the case of mitosis. And in plant cell, you know what is the method of cytokinesis? Yes, of course, it is by cell plate formation. It may or may not takes place. It is not a necessary step. It can happen or it cannot happen also. So, it may or may not takes place after this meiosis I, nuclear division 1. Now, coming to meiosis II. Meiosis II is exactly similar to that of mitosis. So, it is which division? Again it is equational division. Mitosis is known as equational division because the two daughter cells formed after this is qualitatively and quantitatively similar. So, meiosis II, what is meiosis II? Division of nucleus, second division of nucleus in meiosis. So, second nuclear division in meiosis, that is known as meiosis II, and it is exactly the same as that of mitosis. So, it is equal to mitosis. Chromosome number will remain the same, that means haploid. You know in which stage haploid is achieved during meiosis? Yes, anaphase 1, and it is of a shorter duration. What is the longest stage in meiosis? Yes, of course, it is the prophase I. So, this meiosis II, it is of shorter duration and it has again two phases. What will be the phases? Nuclear division, karyokinesis 2 and cytokinesis 2. What is this 2 indicates? 2 indicates? Yes, of course, it is the second nuclear division. Karyokinesis 2, it involves the separation of two chromatids of each of the univalent chromosome. Now, it has different stages, 4 different phases. You know what are the different phases? Prophase II, metaphase II, anaphase II and telophase II. 2 indicates the second nuclear division. Since all these stages, prophase, metaphase, anaphase and telophase, all these phases are belonging to the second nuclear division. So, it is written as prophase II, metaphase II, anaphase II and telophase II. Here you can see the diagram. This is prophase I. Individual chromosomes univalent with the 2 chromatids. This is metaphase I. These individual chromosomes arranged at the equatorial plate. Now, here you can see the separation, anaphase I and telophase I. Prophase II, disappearance of a centrosphere, just the same character as that of prophase. Centrosphere will disappear and centriole will duplicate and they will move to opposite poles. Development of astral rays around these centrioles, and they are the proteinaceous microtubules. Chromatin threads undergo coiling and condensation to form the chromosomes. Chromatin threads we can see during the telophase I, and those chromatin threads, they undergo coiling and condensation to form the chromosomes. So, all these changes that will take place during the prophase II. Each chromosome, when we are observing, they have 2 chromatids because they are univalents. So, they have 2 sister chromatids. They are attached at the centromere portion. See, here you can see these are the 2 cells that we obtained after telophase I, and thus each of these daughter cell obtained after telophase I, they follows this stage of a prophase II. So, this is the prophase II. 2 chromosomes we can see here, that means 2 univalents, and each univalent is having 2 sister chromatids, and these sister chromatids that is attached at the centromere portion. Nuclear membrane starts to disappear, nucleolus will also starts to disappear, and spindle fibers are formed. So, this particular stage, that is prophase II. Now, the next stage, that is the metaphase II, and during this metaphase II, you can see the chromosomes are seen in a highly condensed state. They are the thick and rod shaped highly condensed structures. You can clearly count and see the chromosomes during metaphase stage. Each chromosome has 2 distinct sister chromatids. All these chromosomes, all these univalents, they are arranged exactly at the equatorial plate. That means at the center of the cell. Spindle fiber formation that is completed. So, these are the different stages that you can see here during the metaphase, just similar to that of the metaphase of mitosis. See, here you can see the diagram also. These are the chromosomes, univalents. Then coming to this next stage, continuation point here, each chromosome is connected by chromosomal fibers exactly at the centromere. One of this chromosomal fiber starts from one pole. Centromere of all the chromosomes lie at the equator, center of the cell, and regarding the arms are pointing towards the poles. Arms are pointing towards the poles and centromere is seen towards the center or equator. This is the arrangement of chromosomes here. Now, next you can see the anaphase II. See this case here, diagram, anaphase II. Here during this particular stage, the chromatids of each of the chromosome, univalent, separate. Chromatids are separating, not the chromosomes. Chromatids of the individual univalent separates, and they will move to opposite poles. So, spindle fiber will contract. Contracted spindle fiber will carry the chromatids to opposite poles. That means the centromere of each of the univalent chromosome will split, and the two sister chromatids of each of this chromosome, univalent chromosome, that will separate and move it to opposite poles. So, this phenomenon, that is the anaphase II. So, equal number of chromatids you can see here in each pole. Proteinaceous interzonal fibers. Here also you can see the same thing. I will show you here. Anaphase stage. What happens during anaphase stage is the chromatids, they are separating and moving to opposite poles. Chromosomal fibers, spindle fibers, and here you can see the interzonal fiber. The proteinaceous interzonal fibers developed between the centromere of daughter chromosomes. Daughter chromosomes we will call it as chromatids. Centromere of each daughter chromosome points towards the poles. See, here you can see centromeres are pointing towards the poles and arms are pointing towards the center. We can see equal number of daughter chromosomes in each pole. Here also you can see 3 in one pole and 3 in the other pole. Now, coming to telophase II. See the last diagram here, this is the telophase II. Here you can see how many nuclei you can see here, 4. 4 nuclei we can see here, or even 4 daughter cells we will obtain after this phenomenon. It is just the opposite of a prophase. It is just the opposite of prophase II. Since it is telophase II, it is just the opposite of prophase II. Chromosomes starts uncoiling and decondensation to form chromatin reticulum. Astral and spindle fibers disappears. Centrioles are surrounded by centrosphere, the clear area, and forms the centrosome. Nuclear membrane reappears. Nucleolus also reappears, and we can see one haploid nucleus in each pole, and both are of equal size. In each pole we can see one haploid nucleus in each cell. So, total how many nuclei you can see here? 4 nuclei you can see here, and all are of equal size. Now, coming to cytokinesis II. It is the division of cytoplasm into 2, and in animal cell you know it is by the phenomenon cell furrowing, and in plant cell what is the mechanism of cytokinesis? Yes, it is by cell plate formation. Now, it results in the production of how many cells? How many daughter cells after meiosis? Yes, 4 daughter cells. In each daughter cells, the chromosome number will be reduced to half with that of the parental cell. So, what term we can use for this process? Reduction, division, meiosis. Now, coming to the significance of meiosis. It helps in the production of haploid gametes or even haploid spores. Haploid, what is haploid? With only one set of chromosomes, cells with one set of chromosomes. It maintains a chromosome number of each species constant in each generation. It is only because of meiosis the chromosome number of a particular species remains constant generation after generation. Suppose if there is no meiosis, only mitosis, then what will happen? The chromosome number will be doubling in each generation. So, in order to avoid that doubling, here it is by meiosis the chromosome number remains haploid in the gametes, and by fusion of the gametes again the diploid condition is restored in each generation. It helps in reproduction and it results in genetic variability. Since there occurs the recombination of genes during the Pachytene stage, there occurs genetic variability. It plays an important role in evolution. Since there is genetic variability, it contributes to evolution. Now, see the differences between mitosis and meiosis. What are the differences between mitosis and meiosis? A very important question aspect that you have to be clear about it. See the differences. You can also analyze. Mitosis, we are using the term equational division, and for meiosis it is, yes, reduction division. Why it is known as equational division? You know the answer, because the chromosome number will remains the same. The daughter cells obtained will be always qualitatively and quantitatively similar. And here in meiosis, the chromosome number that is reduced to half. It takes place in the somatic cell, and you know here it takes place in the reproductive cells. Here how many nuclear division? Only one. Nucleus divides only once in mitosis, and nucleus divides twice in meiosis. If it divides only once, how many daughter cells we obtained? 2 daughter cells. Here it divides twice, so 4 daughter cells are produced. It helps in growth, it helps in repair and regeneration. It helps in reproduction. Resulting cells, regarding its ploidy, they may be diploid. If it takes place in the diploid cell, the resulting cells will be diploid, and if it occurs in the haploid cell, then the resulting daughter cells will also be haploid. But here only all the daughter cells will be haploid, and in mitosis the daughter cells will be similar to that of parental cell, and here it will be differing from parental cell because there is genetic recombination, there is variation. And here you can see the prophase that is of a short duration, but in meiosis it is a long prophase with so many substages, 5 substages. Here there is no substage in the case of mitosis prophase, but here in the meiosis the prophase 1 has 5 different substages. So, it is a long stage. No zygote is absent, here zygote is present, pairing of homologous chromosomes. No crossing over, no exchange of chromatids between the homologous chromosomes. Here it takes place. Since there is no crossing over, no chiasma formation. Here chiasma are formed. Now, centromere of the chromosomes lies at the equator here in mitosis, and here in meiosis the centromere is pointing towards the poles. Mainly this will be in the metaphase I stage. Daughter cells divide again. The resulting daughter cells, they can divide again, and here daughter cells do not divide. Now, coming to some of the important questions to solve. Name the following. Pairing between homologous chromosomes takes place in which stage? Yes, your answer is correct, zygote. Crossing over between the homologous chromosomes takes place in which stage? Your answer is of course right, Packaging stage. Describe the following. Synapsis, bivalent, chiasmata. Synapsis means pairing of homologous chromosomes takes place during zygote stage. Bivalent means a group of two chromosomes such as maternal and paternal chromosomes seen during this zygote stage as well as Packaging stage. Now, coming to chiasma, point of crossing over between the non-sister chromatids of a homologous chromosome, that is called as chiasma. Form examples where 4 cells formed in meiosis are equal in size, whereas they are found unequal in size. In spermatogenesis, the 4 spermatozoa formed will be equal in size, but during the oogenesis, ovum formation, ovum is large and polar bodies are smaller in size. Distinguish between anaphase of mitosis and anaphase I. In anaphase of mitosis, chromatids will separate, and in anaphase I, bivalents get separated. Can there be DNA replication without cell division? Yes. In salivary gland cells seen in the Drosophila, there occurs this phenomenon, and it will result in the production of a special type of chromosome that we will call it as polytene chromosomes. Thank you.